Hello everybody, today we're going to talk about the Golgi apparatus. Now, what is a Golgi apparatus? Well, a Golgi apparatus is this organelle that sits next to the rough ER in the nucleus that looks like a stack of pita bread. The main purpose of the Golgi apparatus is to take in proteins, upgrade them a little bit, then stack shipping labels on them and send them off wherever they need to go. So the Golgi apparatus, in a way, is kind of like an Amazon warehouse of the cell. Of course, that's if Amazon upgrade your products instead of just breaking them in the shipping and then refusing to give you a refund. Okay, next let's move on to the endomembrane system, which is what the Golgi apparatus is part of. So the endomembrane system is a system that starts with the nucleus, goes to endoplasmic reticulum and the Golgi, and the main purpose of it is to create proteins. So the nucleus has a bunch of blueprints lying around. These blueprints are all for proteins that the cell needs. Now, the nucleus is all brain and no brawn, so it's unable to manufacture these proteins. So it has to ask some RNAs to go run over to endoplasmic reticulum to deliver the blueprints. So the endoplasmic reticulum looks at these blueprints and thinks, hey, why don't I start making some of these? Now, unfortunately for us, the endoplasmic reticulum is all brawn and no brain. So it could pick up a little blueprint, look at it and say, hey, is that a GAG? Nah, CAC it is. And so these proteins come out perhaps not the way they were originally intended to by the nucleus. So the endoplasmic reticulum will take these proteins, put them in little vesicle containers, and send them over to the Golgi. Now this is where the Golgi's heavy lifting job of carrying the endomembrane system comes in. So the Golgi will take these vesicle containers, crack them open, and take a little peek at the protein that's inside. Once it realizes that the endoplasmic reticulum has screwed up a little bit with the protein, the Golgi will come in, fix it up with some of its tools, maybe even give it an upgrade, if it deems that the protein is worthy of an upgrade. And then it'll put a little shipping label on it and send this protein off to wherever it needs to be. Now this could either be outside the cell or inside the cell. It doesn't really matter. All the Golgi needs to do is put a nice shipping label on it so the cell knows where to send it. Next, we're going to have Walter explain a little bit more about how the Golgi apparatus takes these proteins, upgrades them, labels them, then ships them out to other parts of the body that require these proteins or back to other places in its own cell.
So once the vesicle is sent off from the rough endoplasmic reticulum, it ends up making its way to the cis end of the Golgi apparatus right here, which is at the top of the diagram. And now the way that it receives these packages is the vesicle goes up along the edge of the cis face and then kind of fuses in and then deposits its contents that way. And so it gets deposited into these spots called cisternae, and that's these little folds inside of the Golgi apparatus. And they all do, there's many types, they all have different functionalities, but generally, in most animal cells it gets to about 6. However, it can go anywhere up to 30 cisternae. In some biological cells, that's definitely on the rarer side of things. So what actually happens here is that inside the cisternae is that first of all, things get scanned, products get scanned, and if somehow there's incorrect or missing products, it will send back the contents to the rough endoplasmic reticulum. Hopefully it'll get them right the next time. It's not a big deal. But if it's cleared for that, it's ready to go. What'll happen is it gets a number of processes can happen, such as phosphate groups being added to the proteins, or fatty acids, or removal of monosaccharides. And so once it's done, it puts shipping label on the protein, the finished product, and then it sends it to the transphase, which is at the bottom end of this diagram. And so how it ships it out is the finished product goes to this transphase, and then it kind of slowly buds out and forms another vacuole, and then that will ship to some places like cell wall, where the vacuole will fuse in there to deposit its contents, or maybe they'll float around in the cell, like lysosomes, which are possible products of the Golgi apparatus, or they'll go to another destination, to other organelles.
Thanks for listening to our talk on the Goalge apparatus, also known as the best organelle in the cell. We're excited, and we hope you have a nice day.